Hi, my name is Pushri Aisha Kistina. My metric number is 8205455. Today, we'll be presenting from our understanding based on the article titled Whole Genome Sequencing in Clinical Practice by Bagger et al. Published in 2024. Imagine being able to analyze the entire human genome and identify disease-causing mutations from a single test. This is now possible through whole genome sequencing, or WGS, which has become one of the most transformative technologies in modern genomic medicine. Firstly, the article explained that WGS analyzes the complete human genome, including both coding and non-coding region. This allows simultaneous detection of different forms of genetic variation, such as single nucleotide variants, copy number variations, and structural variants, making WGS more comprehensive than conventional sequencing methods. Secondly, the article highlights that each human genome contains approximately 5 million genetic variants. Because of this enormous amount of data, advanced bioinformatics pipeline and variant interpretation are extremely important to identify clinically relevant mutations. Thirdly, the article demonstrated that WGS provides an average diagnostic yield of approximately 25% in rare disease patients, with even higher diagnostic rates in pediatric and neurodevelopmental disorders. This showed the strong clinical value of WGS in diagnosing previously undiagnosed disease. In addition, the article explains that WGS plays an important role in precision oncology because it can identify actionable tumor mutations, homologous recombination deficiency signatures, and tumor mutational burden that may guide targeted therapy selection. Another important point to highlight in the article is that WGS data can be stored and periodically reanalyzed in the future as new disease-associated genes continue to be discovered. This allows diagnosis to improve over time without requiring additional patient sampling. To better understand this technology, I will now discuss the rationales and objectives highlighted in this article. One major rationale discussed in this article is that conventional sequencing methods may fail to detect important genomic alterations, especially variants located in non-coding or poorly covered regions. Therefore, WGS provides a more comprehensive alternative. Another rationale is the increasing number of patients with rare or undiagnosed diseases. Many patients remain undiagnosed after routine clinical investigations, and WGS improves the possibility of identifying the genetic cause of disease. The article also emphasizes the growing importance of precision medicine. Since treatment decisions are increasingly based on genomic information, WGS has become an important tool for personalized diagnosis and targeted therapy. Next, let's move on to the objectives. The first objective of this article is to explain the clinical applications of WGS, particularly in rare disease diagnosis, pediatric genetics, and cancer genomics. The second objective is to discuss the importance of bioinformatics and variance interpretation in analyzing the massive amount of genomic data generated from WGS. The final objective is to evaluate the current challenges and future directions of WGS, including ethical concerns.
My name is Nur Nasrin Shamil Fatiha binti Mat Hanafi. My matrix number is 8204126. Now, I will continue with the methodology section of this article titled Whole Genome Sequencing in Clinical Practice by Bagger et al., published in BMC Medical Genomics in 2024. The main methodology used in this article is whole genome sequencing, or WGS, which is a sequencing approach that analyzes the entire human genome comprehensively. As you can see in figure 1, the workflow is divided into 4 major stages. First is the wet lab stage. As stated in point 1 is in this slide, blood or tissue sample are collected from patient before DNA extraction and library preparation are performed. The article explains that the DNA is fragmented into smaller pieces and adapters are added to prepare the DNA for sequencing. Next, referring to the point 2 in the slide, the fragmented DNA undergoes NGA-based sequencing. According to the article, fluorescent level nucleotides are used to identify DNA sequence during sequencing. This allows millions of DNA fragments to be sequenced simultaneously. Moving to point 3, the sequencing data is transferred into high-performance computing system, or HPC. As shown in the figure, read mapping and variant calling are performed at this stage. Finally, referring to point 4 in the slide, bioinformatic pipelines are used for variant filtration, annotation, interpretation, and clinical reporting. From what we learned in genomics, WGS is considered powerful because it analyzes the whole genome instead of only selected genes. Therefore, it can detect coding variants, non-coding variants, and structural variation in a single analysis. Overall, this workflow is important because it combines molecular biology technique and bioinformatic approach to support precision medicine and disease diagnosis. Now, bioinformatic pipeline and variant filtering. As I mentioned in the previous slide, after sequencing is complete, genomic data undergoes bioinformatic analysis before clinical interpretation can be performed. The main method discussed in this section is bioinformatic-based data processing and variant filtering. Referring to the left side of the slides, the article explained that the sequencing data first enter FASTQ format, which contains raw sequencing rates. The rates are then aligned to the human reference genome to generate BNN files before genomic variants are stored in VCF files. According to the articles, one WGS analysis can generate around 30 gigabytes of raw data and approximately 5 million variants per patient. Because of this massive amount of data, the article emphasized that high-performance computing system and automated bioinformatics pipeline are extremely important. Now, referring to the filtering graph on the right side of the slide, the filtering process begins with approximately 5 million variants. Low-quality variants and common population variants are removed before prioritizing, coding, and splice variants that may have clinical significance. As stated in the slide, databases such as Genome, ClinVar, and HGMD were also used to compare variant frequencies and pathogenicity information. From our genomic understanding, this filtration strategy is necessary because most genomic variants are actually normal human variation, not disease-causing mutation. Therefore, proper filtering is very important to reduce complexity and identify clinically relevant variant more accurately. Next is SCMG variant classification. After variant filtering is complete, the identified variant are classified using the SCMG variant classification system. As shown in the table in the slide, the article categorized variant into 5 major classes, which are benign, likely benign, variant of uncertain significance or VUS, likely pathogenic, and pathogenic. So referring to the pie chart on the left side, one important finding highlighted in the article is that only a small proportion of variants were truly pathogenic, while many variants were classified as VUS. The reason for this result is because human genome naturally contains millions of genetic variation, and many genes of mutation are still not fully understood scientifically. The article emphasized that VUS represent one of the biggest challenges in clinical genomics because scientists still cannot confidently determine whether these variants contribute to disease development. As stated in the article, additional function studies, computational prediction tools, and clinical evidence are often required to confirm whether a variant is truly pathogenic. From what we learn in genomics, this also shows that WGS not only about sequencing technology, but also about accurate interpretation and understanding of biological significance. Next is clinical application of WGS. Now we're moving into the result section. This slide summarizes the major clinical application of WGS discussed in the article. Referring to the left side of the slides on the major findings, the article reports that WGS has a diagnostic yield of approximately 25% and perform better than exome sequencing. The reason for this higher diagnostic capability is because WGS analyzes the entire genome instead of only coding region. As shown in the figure on the right side, WGS can detect coding variant, non-coding variants, structural variant, and mutation site nature in a single analysis. The article also highlighted that WGS is especially effective for pediatric disorder, neurodevelopmental disorder, and cancer genomics. Another important point emphasized in the article is that WGS support precision medicine by enabling more comprehensive identification of disease-causing mutation. Based on our genomic studies, conventional sequencing method may miss important structural or regulatory variants, whereas WGS provides broader genomic coverage and more complete genomic information. Therefore, this result demonstrates why WGS has strong potential for improving disease diagnosis and personalized treatment strategies in clinical medicine. So for the next slide is precision oncology and AI-based interpretation. As mentioned in the previous slide, one of the major clinical applications of WGS is precision oncology. Referring to the left side of the slide under precision oncology, the article reports that WGS successfully identified actionable tumor mutation homologous recombination deficiency or HRD signatures and tumor mutational burden or TMB scores. These findings are important because they help clinicians select targeted therapies and improve personalized cancer treatment. The reason why WGS is effective in oncology is because it can comprehensively analyze tumor genomes and identify clinically relevant mutation associated with specific cancer therapy. Now, referring to the lower image and AI tool section, the article also emphasized the growing role of artificial intelligence in genomic interpretation. AI tools such as alpha mesons and reverse scores were used to predict whether genomic variants are likely pathogenic or benign. This is important because WGS generates an extremely large number of variants, making manual interpretation difficult and time-consuming. Based on our genomic understanding, AI-driven tools can improve variant interpretation, reduce uncertainty, and support more accurate genomic diagnosis. Overall, the article concludes that WGS has strong potential in future precision medicine because it integrates sequencing technologies, bioinformatics, and AI-based interpretation into one comprehensive clinical approach.
Next, we address the ethical concerns surrounding whole genome sequencing in clinical practice. While this technology is transformative, it raises significant issues regarding privacy, confidentiality, consent, and the psychological stress placed on patients. It also impacts biological relatives and brings up risks of social stigmatization, insurance, and employment issues. On the left, we look at the challenges inherent to the process itself. Because of the vast amount of information generated, there are three main concerns. First, patients may not fully understand the long-term implication of their data. Second, because the data is permanent and complete, it is difficult to foresee future applications or the dilemmas patients might face years down the line. Therefore, providing in-depth information to the patient prior to analysis is absolutely critical. On the right, we see the risk associated with privacy and data sharing. Since data management is often involves third parties, we must prioritize data storage and the prevention of any unauthorized use of patient information. Proper guidelines and thorough education for the staff involved. Ultimately, we must ensure patients are comfortable with the technology so they can be properly informed. Next, I'll be explaining the advantages and limitations of WGS, which is whole genome sequencing over other methods. Firstly, almost half of the variants identified in recent UK and Ireland rare pediatric disease were WGS study. It detects more variants not only in the large non-coding parts of the genome but also in exons due to a superior mapping quality. WGS captures copy number variations and structural rearrangements as well as mutation signatures and polygenic scores. WGS can be considered a lifelong investment that may be revisited for different clinical purposes and reanalyzed when novel pathogenic variants and disease-causing genes emerge, and also be used as personalized medicine. On the right-hand side, limitation of whole genome sequencing over other methods, which requires a robust computational infrastructure to ensure fast and reliable data processing. WGS pipeline must fulfill a set of requirements concerning both the physical computational and the software application infrastructure. And the analysis is about 30 GB. The variant files of WGS come near 1 GB, which corresponds to an increase in data of 13.0, which is 24-fold respectively. Recent risk sequencing machines with built-in data processing hardware and close to end-to-end workflow may also bring limitations on how to reprocess samples and Next, we address the ethical concerns surrounding whole genome sequencing in clinical practice.
While this technology is transformative, it raises significant issues regarding privacy, confidentiality, consent, and the psychological stress placed on patients. It also impacts biological relatives and brings up risks of social stigmatization, insurance, and employment issues. On the left, we look at the challenges inherent to the process itself. Because of the vast amount of information generated, there are three main concerns. First, patients may not fully understand the long-term implication of their data. Second, because the data is permanent and complete, it is difficult to foresee future applications or the dilemmas patients might face years down the line. Therefore, providing in-depth information to the patient prior to analysis is absolutely critical. On the right, we see the risk associated with privacy and data sharing. Since data management is often involves third parties, we must prioritize data storage and the prevention of any unauthorized use of patient information. Proper guidelines and thorough education for the staff involved. Ultimately, we must ensure patients are comfortable with the technology so they can be properly informed.
In conclusion, this article clearly demonstrates that whole genome sequencing has become one of the most impactful technologies in modern clinical genomics. Firstly, WGS significantly improved diagnostic accuracy by enabling comprehensive detection of disease-associated genetic variants in both rare diseases and cancer. Secondly, the article emphasizes that bioinformatics and variant interpretations are essential components of WGS because the technology generates massive amount of genomic data that require careful filtering and clinical evaluation. Thirdly, despite its enormous clinical potential, important challenges still remain, particularly regarding ethical concerns, computational infrastructures, data storage, and interpretation of variants of uncertain significance. Finally, the authors conclude that continuous advancement in sequencing technology, artificial intelligence, and multi-omics integration will further expand the role of WGS in precision medicine and personalized healthcare. Overall, this article highlights that whole genome sequencing is not only improving our understanding of human disease, but is also shaping the future of modern medicine. That's all from all of us. Thank you so much.